Well, thank you very much, Katya. I'm overwhelmed. I don't know if I can quite live up to all that. And I'm really delighted to have this invitation, the more so because I reminded the organisers that for some time now, my work hasn't really been primarily involved in 22q11 research. Rather, it's been involved in spin-offs from research we've done into 22q. And so I've been given license to give you a little bit of history to my involvement in the syndrome as a kind of light introduction into what I hope you will agree with me is a bit of unfinished business on 22q. And then finally, I will describe one of the spin-off projects that we've been involved in to put some scientific meat on the bones of this presentation, if you will. So basically in 1985, as Katya has mentioned, I was working in Bob Williamson's group at St Mary's on cystic fibrosis. And in 85, we and other groups showed that the cystic fibrosis locus was closely flanked by an anonymous marker D7S8 and the proto-oncogene MET. And then serendipity reared its head because we just had a seminar in the department on the creation of somatic cell hybrids. And one of the selection mechanisms for specific chromosomes was the presence of an active oncogene. And just the year
Dr. Scambler, do we see your slides?
Do you not see them?
I don't think so.
Oh, we've been, okay. I've got them on my 1 second. I'm sorry about that. Let's try again. Obviously the sharing didn't work.
Yes,
second.
now it does.
Is
you
that
go
better?
in full-time screen sharing mode. Super.
Still see them?
Yes. Oh, now we can see your
okay.
presentation.
Somehow it must have unactivated the share.
You
okay.
may want to go into the icon that goes into full presentation mode.
Is that what do you see?
We're seeing your presenter view.
Okay. Okay, so let's just try.
Go here.
How about now? You okay?
Yes.
Yes, that's great.
Okay, right. So the year previous to the linkage, a guy called Colin Cooper working in George van der Voede's lab had molecularly isolated a mutant form of MET, which was the oncogenically transforming version. So the other piece of serendipity was Colin had just flown over the Atlantic to start his own lab in London. So I was very quickly on the phone and within a few weeks was in his lab. The challenge we had was to adapt his transformation technique, which used small sheared DNA fragments to whole chromosomes. And the technique of chromosome-mediated gene transfer is actually quite simple. All you do is grow your tumor cells, arrest them in metaphase, spin down the chromosomes, and then use calcium phosphate together with various other alchemic tweaks and sprinkle them onto NIH3T3 cells. And then you wait, and if you're very lucky, we worked for several months, you get foci, and then you can expand those clones. And we eventually isolated a number of clones that contained a few megabases flanking the cystic fibrosis locus. Unfortunately, despite these hybrids, we were only able to isolate the gene next door to cystic fibrosis, close but no cigar, as they say. Now, the reason for telling you all this is that I thought that for the time in human genetics, this particular technique was very powerful and could be applied to all sorts of other regions of the human genome, but where to focus? I was aware from attending clinics at Great Ormond Street Hospital of this thing called the DiGeorge syndrome, and that the late Frank Greenberg had described the syndrome in association with visible cytogenetic deletions of 22q11 and possibly also at 10p13. So by analogy with contiguous gene deletion syndromes like Wilms tumor, I wondered whether DiGeorge could also have submicroscopic deletions. Now, also at the GOS clinics, I came across a patient who had the so-called DER22 syndrome or Emmanuel syndrome, which essentially these patients have an extra copy of genes on proximal 22q11 and some on distal 11. The phenotype of these patients is very similar to that seen in the dicentric chromosome 22 of cat-eye syndrome. So this region of proximal 22q seemed particularly rich from the point of view of a budding genetic dysmorphologist. Similarly, so did 9q34, where 3 different syndromes had been mapped by genetic linkage by the mid to late 80s. Now, what you may think do 22q11 and 9q34 have in common? The answer is the Philadelphia chromosome, which is the fusion between the breakpoint cluster region, which drives expression of an able fusion oncogene. So by now, you will have guessed what Scambler was thinking. Use the Philadelphia chromosome in CMGT experiments, create hybrids, and solve goodness knows how many different genetic problems. And indeed, the BCR-ABL fusion is capable of transformation. But what the young man didn't know at this point was that it does not transform NIH3T3 cells. So what could have been a futile 2 years or so was rescued by a conversation down in the bar. What else? There was a new recruit to the Williamson empire who was reneging me with stories of his previous lab at the Karolinska Institute, and in particular about this young PhD student called Jan Dumansky. And he was mapping locus for meningioma that was in the middle of the long arm, and essentially he was shoving all the probes proximal to his region of interest at the back of a freezer drawer. In other words, DNA segments that I might have been particularly interested in. So once I picked up the phone and was soon in Stockholm having very nice conversations with Jan. It wasn't all phonoclone though, and we did have some fun using then novel techniques to isolate our own. And my very first PhD student, Alison Carey, used a technique called microcloning to isolate more DNA probes from 22q11. And here is a picture of some metaphase spreads where you can see she has scraped away our region of interest here and isolated some microclones for 22q11. So armed with a battery of markers, we, in fairly short order, were able to show that, yes, indeed, many cases of DiGeorge syndrome and similar dysmorphologies were caused by submicroscopic deletions within 22q11, and moreover, that they could be associated with familial DiGeorge syndrome, the corollary of which is that the vast array of expressivity and penetrance that we are now very familiar with in this condition. However, look at the journal Genomics, and 5 years later, what a difference. The vanishingly rare chromosome 10p deletion found its way into Nature Genetics. The vicissitudes of science for you. Now, so as well as pursuing a shortest region of deletion overlap mapping strategy, another strand that we pursued involved a unique family that had been reported back in 1986 by a French group in this one-page report. And this family segregated a balanced 222 translocation. And for those of you around in those days, such balanced translocations were gold dust, because it was commonly assumed that balanced translocation breakpoints would interrupt major genes involved in genetic disorders, and cloning the genes at the breakpoint would help you identify said genes, wouldn't it? Well, this is 1993 now, and we thought we had it. And what you can see on the left is a metaphase spread from the proband in the ADU family from France. And you can see signal on the normal 22, and you can see signal on both derivative chromosomes, suggesting that this particular cosmid, which was identified by one of those microclones, spanned the balanced translocation breakpoint. Now, this on the right-hand side is what the signals should have looked like in interface preparations, with a single nice signal for each of the chromosomes 22. And this is what the cosmid on the left looked like. And you can see all the several signals that proved or indicated that we had identified low copy number repeat sequences that were specific for 22q11 and flanked the locus. Now, I was so disgusted with what evolution had dealt us here, I dropped this entire piece of work on low copy repeats. And that was an absolutely stupid decision, because as you're probably aware, Bernice Morrow, Beverly Emanuel, and Yoresh Ramesh, and we'll hear from him later, have gone on to do fantastic work in this area. But there were some other longer-term benefits from this particular piece of research, because it's our first publication along with Antonio Baldini, who taught my group fluorescence in situ hybridization, first at Yale with David Ward, and subsequently when he came to London at the ICRF laboratories, where Elizabeth Lindsay, then a postdoc in my laboratory, carried on the collaboration. And to misquote Emily Bronte's Jane Eyre, viewer, she married him. So another 3 years on, and we did indeed, despite the repeats, manage to get to the balanced translocation and isolate a gene that was disrupted by this chromosome aberration. Unfortunately, this gene proved a bit of a nightmare. It didn't encode for any long open reading frame, and it had many alternate splice forms. And we couldn't find any de novo mutations in the patients that we'd sequenced. So we were getting very frustrated and went on to study other genes. However, I think that it may be worth returning to this gene. This is the unfinished business, simply because since we published this, the DGCR5 has been shown to be functional. And in the 10 years since we published this paper, there were just 3 publications that mentioned DGCR5. Not great for one citation index. In the subsequent 5 years, there were 43, predominantly because of DGCR5's role in various cancers. But there are also, in addition, some interesting papers. One involves REST, the repressor element 1 silencing transcription factor. And what was already known, that this transcription factor regulates various genes involved in the central nervous system. And what this paper added was that REST regulates a good proportion of long non-coding RNAs, and the DGCR5 was their exemplar of a gene expressed in the central nervous system and a direct repressed target of REST. But more provocative, perhaps, is the more recent paper. And this used bioinformatics to identify gene networks where DGCR5 turned out to be a hub within networks involved in neuronal function and in schizophrenia. So they then went on and did some loss of function and gain of function experiments to show that DGCR5 is involved in these networks. So perhaps then DGCR5 is something that has passed a lot of people by and may be worth revisiting. And it could well be amenable to the sort of work in organoid cultures that many of you are now using. So, as I've mentioned, much of my work has been involved in projects that have spun out of TBX1. But since TBX1 emerged as the major candidate for the disorder, we've been involved in 3 areas. First of all, what are critical downstream targets for the haploinsufficiency as well as the null phenotypes? And it's this first area that I want to dip into next. And the work I'll describe featured prominently Sarah Ibens, a postdoc in the group, and she conducted transcriptomics, which first showed that CXCL12 was downregulated in TBX1 mutant pharyngeal arch dissections. And she then validated this by in situ hybridizations as wild type null. And in particular, what we were searching for were genes that encoded ligands that would communicate between the pharyngeal surface ectoderm, you see the L12 expression here, absent in the TBX1 mutants, which would communicate with the underlying neural crest as it migrated to the arches in the outflow tract. And the receptor for CXCL12, which is called CXCR4, is indeed expressed in neural crest, albeit in a relatively small number of that cell type. In addition, the L12R4 system is known to be involved in neural crest migration in Xenopus and in lateral line and primordial germ cell migration in zebrafish. So we felt that this was a good candidate. Let me now just introduce the pathway a little bit for you so you can understand better what's coming next. So the ligand, as I've said, is CXCL12. It binds the CXCR4 G protein coupled receptor, and that leads to usually a protein phosphorylation cascade and downstream cellular functions. The receptor is then phosphorylated by GRKs, which leads to endocytosis and either degradation or recycling. Now, there is another receptor for CXCL12 called CXCR7, which can either antagonize CXCR4 signaling by scavenging, work with CXCR4, or signal off its own accord. So just one other thing to point you in the way of is that CXCR7 is otherwise known as atypical chemokine receptor 3, and CXCL12 is otherwise known as STF1, just in case I switch nomenclatures. So this pathway certainly has its fans, let's say, and they like to point to the richness of the pathway and the exquisiteness with which it's controlled to affect different pathways within the same tissue. And also, they claim it's the only chemokine system where survival to live birth is important. Okay, these acolytes do have a point. These are just some of the non-cardiovascular roles of the CXCR12, CXCR4 pathway. And many of them actually involve neural development or regulation within the immune system. Within the cardiovascular system, we were actually quite disappointed when we set up our genetic interaction experiments, because basically, no matter how many partial loss of function alleles we combine with TBX1 loss of function heterozygosity, we couldn't get an interaction. However, the pathway is certainly important for development of the cardiovascular system, including some of the developmental processes that are also affected in DiGeorge syndrome. So we may learn something. And perhaps the most important discovery we made, it was that CXCL12, CXCR4 were both important for development of the coronary arteries. And you can see here the ventricular wall and the smooth muscle outlining normal coronary arteries in the wild type, which are completely absent in the null embryos. And what you can also see in this next panel here are the nicely formed semilunar valves in the aorta. And here in the CXCL12 nulls, you can see that they are dysplastic. Now, Sarah went on to actually quantitate this using 3D reconstructions of confocal images, and they're shown here. So this is a particular florid example of malalignment of the valves. So you can see in the wild type how they're basically on the same level, whereas here we have one valve piled on top of the other two. You've got a frontal view and an overhead view for you there. So we thought that this was maybe a phenomenon worth investigating further. The reason being that valve defects are actually pretty common. So 1 in 200 people are born with a bicuspid aortic valve. And by the age of 56, I think it's about 2% of Americans will have aortic stenosis. So pathways involved in valve development can possibly be re-invoked in valve repair, regeneration, tissue engineering, and so on. So we thought we'd add to that mix. To take you a little bit further, let's look at some of the processes involved in semilunar valve development. So the semilunar valves develop within the outflow tract here in the cardiac cushions. And these are essentially accumulations of extracellular matrix sandwiched between the endothelium or endocardium of the outflow tract and the muscular wall. Okay. Now, during a very important process called endothelial to mesenchymal transitions, the endocardial cells lose their apicobasal polarity and migrate into the underlying ECM, adopt a front-back polarity, and lay down mesenchyme and help sculpt the developing valves. Another contribution comes from the neural crest, and this is required for normal valve development as well. Okay, so we then looked at the expression of CXCL12 and CXCR4, and what we can see is that CXCR4 is largely expressed in the endocardial layer, and the cells that have just undergone endocardial to mesenchymal transition just underneath the endocardial surface there, with reciprocal CXCL12 expression in the surrounding cells. So we then did a lineage trace using Ty2-Cre, which will mark cells that are endocardially derived. And we found that the majority of cells that express CXCR4 within the underlying mesenchyme were actually derived from Ty2-Cre, although there was a small contribution from non-endocardial second heart field and also from the neural crest. So this is a lineage trace with PAX3-Cre, which is a neural crest marker, and it just shows you at 11.5 just how few cells actually express both the reporter for neural crest and the CXCR4. And CXCR4 is actually activated in neural crest derived cells later during development. Okay. Oh, so the eagle-eyed amongst you will have perhaps spotted that the spots, this punctate nature of CXCR4 expression, which you can also see here. And if we look at CXCL12 nulls, what we see is that the CXCR4 antibody no longer detects these puncta, but instead is located on the membrane. And moreover, the expression seems to be away from the lateral edges of the endocardial surface and more to the medial part of the developing valves. And the cells are kind of clumped together, suggestive of abnormal migration of the endocardial cells post-endo-MT. So we next did a lineage trace to check that out. And again, what we've got here is a kind of florid example, but in a CXCR4 heterozygote, you can see the trace cells are pretty evenly distributed between each of the semilunar valve leaflets. Whereas in the absence of R4 signaling, in this example, almost all the cells have found their way into this right leaflet here. And of course, as these cells proliferate, you will then get dysmorphic valves, which will probably fail to co-act and be incompetent. Okay, so another important thing about this punctate expression is that by E12.5, it's virtually all disappeared. I mean, maybe there's a little bit of spottiness here, but by 14.5, it's gone. And if we blow up that signal there, you can see it's very obvious, the puncture. But if you blow up a region of the E12.5 wild types, you can see it's virtually all membranous. So that we feel that there's a very important switch in how CXCR4 is behaving in wild type embryos over this 24-hour period. So where were these puncta within the cells? That was our next question. So we used a series of intracellular organelle markers and showed that the puncta co-localized with the Golgi and late endosomes, which was of interest to us because in migrating cells, it's often the case that the Golgi apparatus become polarized with nucleus posterior, Golgi anterior. So we felt we could use this as a kind of surrogate for detecting migrational direction in vivo as a snapshot. And this is indeed what we did, comparing wild type embryos with CXCL12 mutants. And in short, essentially, in the absence of L12R4 signaling, movement away from the endothelial surface is randomized, supporting the idea that there's abnormal migration in the absence of this signaling pathway. Okay, so to corroborate this and to be able to do a little bit more in the way of biochemical analysis and various other factors you can see on the slide here, we wanted to adopt an in vitro system as well. And in addition, we felt that this would help with some live cell imaging. And Liam Ridge within the lab spent some time adopting a new procedure. So what he did was took 10.5 day embryos, dissected the outflow tract, and then teased them open. He laid them down, endocardial surface downwards, onto the collagen gel within a modified Boyden chamber. Then the cells were allowed to, the endocardial cells were allowed to enter the collagen gel over a 72-hour period, so that the effects of adding a CXCL12 gradient or not at 1 and 4 hours, so a fairly acute response, could be monitored. And essentially what we showed from these experiments was that over a 1 hour period, CXCL12 did indeed induce a fairly rapid orientation of cell migration away from the surface of the gel, and that this was dependent upon CXCR4, as shown by the CXCR4 antagonist AMD 3100. Of interest, the endocytosis inhibitor dinosaur also abrogated this response, showing that internalization is likely required for this signal response. And in terms of signaling pathways, the MEK pathway failed, inhibition of the MEK pathway failed to alter this migratory response to L12, whereas PI3 kinase inhibition did. So PI3 kinase seems to be the operative pathway. So given the likely importance of this switch from punctate to non-punctate expression indicative of high levels of R4 signaling, we decided we'd do a timed knockout of the receptor. So we did this using a universally active tamoxifen responsive Cree transgenic mouse, and we activated the Cree at 13.5, so well after the punctate should have disappeared. And what we found was that, first of all, that the Cree was fairly effective at reducing CXCR4 expression in the conditionals, and expression has virtually disappeared by the time you get to 18.5 days. And second, that the valves seem to have formed pretty much at the correct place vis-a-vis one another. So they're not piled on top of one another. There's little in the way of malalignment, but what there is hyperplasia, which suggests that there is a late requirement for CXCR4 in controlling cell proliferation. So examine cell proliferation using EDU marking. And you can see the results here. So I'll just walk you through. At 12.5 days in CXCR4 positive mesenchyme, there is incorporation of EDU at about 37% levels, and this drops at E15.5. So proliferation is downregulated in that time window. However, in the absence of CXCL12, this downregulation of proliferation fails to take place, which is consistent with our ideas about the valve hyperplasia. But also of interest, you can see that even in the CXCR4 negative mesenchyme, there is also a decrease in proliferation. So other pathways are obviously active, but also of interest is that even these pathways in CXCR4 negative cells are affected just about significantly in CXCL12 mutants, suggesting some non-cell autonomous effects from the CXCR4 positive cell population. So what about the other receptor, CXCR4?
Dearest Dr. Scambler, we are almost on time. I think
okay,
our timers on Zoom.
This is my pre-summary.
Super.
Okay. So basically in CXCR4, 7 mutants, the key is that again, we see hyperplasia, but very little in the way of malalignment. And if you look at 15.5 day images, you can see maintenance of puncta, suggestive of ongoing CXCR4 signaling, and that CXCR7 acts as a sequestrator in this particular context. So in summary, therefore, in E11 wild type cells, we have high levels of signaling through the PI3 kinase, and during development, L12 levels drop, R7 goes up, we have lower levels of R4 signaling, we lose the puncta, and in the absence of CXCR7, we maintain the puncta and we maintain the high level signaling, and we lose the low level signal, whatever the second messenger is, that's required to control proliferation. So this is perhaps relevant, as I mentioned earlier. People are already using CXCL12 in tissue engineering experiments, but it would be nice to end on something more relevant to 22q. And this just draws attention to the role of this chemokine pathway in the neurovascular unit, and in particular in interneuromigration We may hear something about this from Anthony later, but it's just to show you that the R4L12 signaling pathway has already been implicated in brain and behavior. So I think that this is a pathway, again, something that is worthy of more analysis by the community. So I have to finish by acknowledging the people that did the bulk of the L12R4 work. That's Liam Ridge and Sarah Ivins and the funders, and you all for listening and allowing me to indulge in a little bit of history before going on to the science. And just to remind you, I hope to see you all in Croatia in June next year. Thank you very much.
Wow, this was a fantastic talk. It felt like a front row seat to a time lapse right through history. Dr. Scambler, there are a million questions, including from myself, because there are so many parallels to the thymus, but in the interest of time, I'm going to lead by example, and we keep the questions for either in the end or for Croatia. And I'm turning it over to Dr. Donna McDonald-McGinn, who of course you all know, who is leading the 22q11.2 Society and is also a scientific advisory board member of our Stanford Hamilton U thanks to 22q11 Deletion Syndrome Scientific Advisory Board. Donna and Dr. Scambler, thank you so very much.
Thank you.